Hi, good evening, and thank you for joining me on my presentation for my research proposal. Before we get started, I would like to just introduce myself and give you a little background. My name is Rosalind Dudding. I am in the Navy. I've been in the Navy now for 17 years. I'm currently stationed at the Naval Medical Center Camp Lejeune, where I'm the leading chief petty officer for the laboratory and blood services departments. Um, when I came into the Navy, I came in as a hospital corpsman. And as a hospital corpsman, you learn uh, lots of areas in the medical field so that you're able to assist nurses and doctors and physician's assistants in any way possible. In 2005, I was sent to Iraq, and one day a sailor was badly injured, and he came into our surgical shock trauma unit, which is kind of like an ER, and he was very badly injured, and he needed whole blood. So they called over to the birthing area, and they asked for anybody who was A positive to come over to the hospital and donate blood. And I happened to be in the right place at the right time and the right blood product, or the right blood type. And I thought it was amazing to be able to donate blood, have them tested across the room, and taken down the hall to help save someone's life so they could get to that next echelon of care. And at that moment, I had my life-changing event where I said, this is what I want to do with the rest of my life. So in 2007, I graduated with my associate's degree from Thomas Nelson Community College as a medical laboratory technician. And since then, transfusion services, the blood bank, it has been my passion ever since, which now brings me to my research proposal of cold storage platelets in 100% platelet additive solution for 25 days as being the new standard storage method. I have a few handouts if you would like to read along, and also my contact information is on there as well. So since 1969, the standard of storage for platelets has been in room temperature for 5 days under constant agitation. This prevents changes in pH, bacterial growth, and platelet degradation. Since then, the FDA has actually approved storage of apheresis platelets for 3 days at temperatures 1 to 4 degrees Celsius, which is huge. We have so many other blood products, and by far platelet has the shortest shelf life. So for many years, platelets were originally suspended in a percentage of its own plasma in order to preserve its lifespan prior to transfusion. Since then, platelet additive solutions have replaced plasma and become the new storage media for plasma, because plasma contains enzymes that negatively affect the quality of platelet concentrates. So these additives, they contain electrolytes, and that allows us to modulate how platelets behave in terms of metabolism. So what they have found now is potassium and magnesium containing platelet additive solution has now proved to be the best the best solution for platelet quality and survivability, as well as it even reduces the amount of allergic reactions and transfusions. So what if we were able to combine multiple storage mechanisms along with maybe a different additive solution for platelets? So packed red blood cells currently have a shelf life of 42 days, depending on what type of additive solution you use. So these additives preserve erythrocyte function without decreasing the amount of red blood cells prior to transfusion. So by eliminating plasma suspensions of platelets and creating an additive similar to those of packed red cells, we could store our platelets at 1 to 6 degrees Celsius. We could extend the shelf life from 5 to maybe 25 days. So for this proposal, it's going to be purely experimental. We'll have apheresis platelet donations collected at the Naval Medical Center of Camp Lejeune by our Blood Donor Center staff. We'll follow all local standard operating procedures because they all fall in line with AABB and CAP, as well as our Navy blood program. The donations will be collected using our Fenwal amicus separator with corresponding amicus apheresis collection kits. Then we're going to have two groups. We'll have a control group and a test group. So our control group is going to be the standard storage that it is now, 65% platelet additive solution in a 35% plasma suspension and stored at room temperature and still under constant agitation. Our test platelets, however, are going to be stored in 100% platelet additive solution and refrigerated at 4 degrees in our awesome Helmer reefers that we have. Of course, we'll have to get donor approval because these donations will be quarantined and they will not be utilized. So we will definitely have to have all of our participants, sign a waiver saying that they do understand that. So what we'll do in the beginning is we will test all of our samples to make sure to get us a starting point for pH, as well as our platelet count, making sure that we don't have any bacterial contamination. And we will use our own microbiology department to test our samples for bacteria. Additionally, we will test for partial pressures of carbon dioxide, oxygen, glucose, and lactate using our iStat blood analyzers. The only thing that we currently don't have that we would need to purchase for additional equipment is going to be our platelet aggregometer to analyze platelet morphology and aggregation formation. We will use a total of 10 donations and during the beginning stages of our research, and this is just in the event that we have any equipment malfunctions or we need to repeat our testing. The refrigerated and room temperature stored apheresis platelets will be tested on days 1, 5, 7, 14, and 25, and we will compare each accordingly delineating any changes in volume, platelet count, pH, and the presence of any bacteria. For this study, all of our donations, again, they will be considered quarantined and not available for transfusion. And we'll use our Wizard Pro for Mac. It's going to be our statistical software used to collect and organize all of our data. For our expected results, I do expect that past the 7 days, since we don't use them in this capacity, that we are going to have a lessening of the content, maybe a slight increase in pH and PCO2 and O2. But in previous studies, all of these results have been pretty comparable and still able to be transfused. So in conclusion, studies have reviewed for this that I reviewed for this research were congruent in determining that platelet function is superior when stored at colder temperatures and in more platelet additive solution than the regulated 65%. Once shown that the longevity of platelets can be preserved, this will increase the apheresis platelet supply and decrease the recurring need for apheresis donors. So thank you again for your time. My contact information again is on, and I truly look forward to working with you on this research.